A lab in Denmark built a snake antivenom out of 8 molecules. 8. Each one has a name and a written sequence you could send to a factory as a file. Tested against the venoms of 18 of Africa's most dangerous snakes, those 8 stopped 17 of them from killing mice. None of it has been in a person yet, and the distance to a rural clinic is the last act of this story. What the 8 already prove is something the field thought was out of reach. The thing they replace is a horse. You inject a horse with venom over months, take its blood, and purify the antibodies it made. The method? Unchanged since the 1890s. Nobody can say exactly what ends up in the vial, and in some products, as little as 1/10 of the protein neutralizes venom. The rest is horse. Nobody had built a defined recipe because venom looked impossible to pin down. Dozens of proteins in a single venom, and an assumption that only a whole animal's immune system could cover them all. The Danish team asked it backwards. How few antibodies are actually enough? The work comes from Shireen Ahmadi and colleagues in Andreas Laustsen's group at the Technical University of Denmark, published in Nature in October 2025. Their answer starts with a piece of accounting. Venom is chemically complicated. The part that kills you is not. From proteomic profiles of all 18 venoms, the team cut the target list to 7 toxin families. Cover those 7, and you have covered a continent's cobras, mambas, and rinkholes. 3 of those families attack the junction where a nerve tells a muscle to contract. Alpha neurotoxins sit down on the acetylcholine receptor, the switch that carries the order. The nerve keeps firing, the receptor never opens. Muscles go slack. And if it reaches the diaphragm, breathing stops. The Mamba toxins work the opposite way. Kunitz dendrotoxins block potassium channels on the nerve side. Potassium is how a nerve resets after firing, so plugging those channels leaves it stuck in an excited state, dumping signal. Paralysis on one side of the synapse, overload on the other. Then there is the damage where you were bitten. Cytotoxins wedge into cell membranes and take them apart. Phospholipase A2 cuts the lipids those membranes are built from. Together, they kill tissue, and that is what sends people to surgery. To get binders against that list, the team immunized 1 alpaca and 1 llama with all 18 venoms. Camelids make a second kind of antibody with no light chain at all. Its whole binding site is a single small domain that works alone, so you can lift that domain out and use it. Those domains went into phage display libraries, where each virus particle carries 1 nanobody on its surface and the gene for it inside. Wash that library over purified toxin fractions, keep only what sticks, and use less toxin each round until the weak binders fall off. 3,000 were tested. 21 bound across several species at once. 8 made the cut. They did not mix all 8 and hope. They built the cocktail 1 molecule at a time, and every step was a live or die mouse experiment. 2 nanobodies first, 1 against short-chain alpha neurotoxins, 1 against long-chain. That pair alone protected every mouse against Egyptian cobra and forest cobra venom. Green mamba venom needed more. It also carries aminergic and orphan group 11 toxins, 2 three-finger subfamilies with no clean textbook receptor story, so 2 more went in. 4 now, and 4 was enough for that snake. Then black mamba. The 4-part mixture kept failing. Black mamba venom is loaded with Kunitz dendrotoxins, and nothing in the mixture touched them. A 5th nanobody went in against that family, and the mice lived. They also ran it in reverse. Strip the mixture back to 3, challenge it with Jameson's Mamba, and most of the mice died. Every entry earns its slot. One more module covered the tissue damage. Against black-necked spitting cobra venom, 2 anti-cytotoxin nanobodies plus 1 against phospholipase A2 protected every mouse. Add that to the neurotoxin core, and the list closes at 8. The full test is the World Health Organization's standard potency model. Venom and antivenom mixed in a tube, held at body temperature for half an hour, then injected together. 3 times the lethal dose, 5 mice per group, survival read at 24 hours. 17 of the 18 came back with every mouse alive. The one that did not was the eastern green mamba, where the cocktail delayed death without preventing it. The authors suspect toxins outside the 7 families, acting together. Then the harder version. Inject the venom first, wait 5 minutes, and only then give the antivenom through a vein. Across 11 venoms, 7 were fully protected. Forest cobra partly. Black mamba and Jameson's mamba only had their death pushed back, and the eastern green mamba got nothing. Against a commercial product, Inoserp PAN-AFRICA, a horse-derived antivenom made for these snakes, the mixture did better on survival and tissue damage for every species except black mamba. One caveat the paper states plainly: the batch they could get was expired because supply was scarce. The size is doing real work. A whole antibody runs about 150 kilodaltons. One of these nanobodies is 12 to 15, small enough to leave the bloodstream and reach tissue a large molecule struggles to get into. That let them try something the conventional product cannot. They injected cytotoxic venom into the skin, waited 15 minutes until the tissue was already discoloring, then injected nanobodies into the same patch. 3 days later, the lesions were significantly smaller. Tissue death drives the amputations, more than 10,000 a year in sub-Saharan Africa. And because a nanobody is one small protein sequence, it can be grown in bacteria. Defined genes, a fermenter, a blend at fixed ratios. No horses, no venom farm, no batch that quietly differs from the last. The gap between that and a person in a rural clinic is wide. Start with what is not covered. These 8 are built for elapids, cobras, mambas, the rinkhals. Africa's vipers are untouched. Puff adders and saw-scaled vipers cause a large share of the continent's bites, and their venom kills by a different route, enzymes that wreck clotting and damage the kidneys. That is a second target list, and nobody has drawn it up. It is also all mice, and mouse pharmacokinetics are not human pharmacokinetics. But this is the one that bites hardest, the clock. In the rescue experiments, the antivenom arrived 5 minutes after the venom, 15 for the skin lesions. Someone bitten in a rural district is often hours from treatment. Nothing here says what happens at hour 4. The short half-life is a real design problem too. Being small is what lets a nanobody reach tissue, and it is also why the kidneys clear it quickly. Venom can keep leaking from a bite site after the nanobodies are gone, and making them bigger hands back the tissue penetration that made them worth using. That trade-off is the likeliest explanation for the black mamba result. Mixed in a tube, the cocktail neutralizes black mamba venom completely. In a living animal, 5 minutes behind the venom, it bought the mice 90 extra minutes, and then they died anyway. What the paper establishes is narrower than a cure and more interesting than an incremental result. For more than a century, antivenom came from animals because of a belief about complexity, that venom was too varied to write a recipe for. 8 named sequences against 17 of 18 venoms? Is that belief coming apart? Their next step says how they read it. Before any human trial, They want the pharmacokinetics in a large animal, a sheep or a pig. And they want to know whether the list can get shorter than 8. The question is no longer whether venom can be covered by a defined set of molecules. It is how small the set gets.